Hi, I'm Anzel Bulofse. Student number is U25572522, and I did the illustrating and part of the editing.
Hi, my name is Donatella Mauldin. My student number is U24998037, and I was filming and I helped edit.
Hi guys, my name is Hope Litzori. My student number is U25308883. My job was script writing.
We chose option explainer.
After that extremely awkward intro, let's just watch me doodle and talk about the prenatal development. There's a few myths about the emergence of a new life. Human beings begin their life inside the womb as a miniature person, complete in every respect and fully functioning. The truth is that during the prenatal phase, the person simply grows bigger and bigger until he is ready to be born. To understand prenatal development, we have to discuss its stages. The first stage being conception.
So, let's talk about conception. Human development begins within a fraction of a second in one of the two fallopian tubes in the woman's body. The egg travels along the fallopian tubes in the direction towards the uterus, which is also called the womb. The eggs, or the ova, are the biggest cells in the body, which are roughly the size of a quarter of a full stop that you see at the end of a sentence. During ovulation, which takes place approximately every 28 days in the reproductive female, an egg is released. It takes the egg about 12 hours to reach the middle of the tube, and at this point, the egg is met with the sperm. A male sex cell and a female sex cell unite. More specifically, the sperm penetrates the eggshell, which is called the process of fertilization. Fertilization is possible as long as the sperm cell stays alive, and its lifespan is 5 days. An egg remains ready for fertilization for approximately 24 hours. If there's no sperm that meets up with the egg cell begins to break down in the uterus. The sperm, which looks very much like a tadpole when viewed under a microscope, is one of the smallest cells in the body, whereas egg cells are already presenting in the female at birth. Sperm cells produced in the testes of a reproductive male get produced at the rate of approximately 300 million per day. The egg cells and sperm cells are both called gametes. The fertilized egg is called a zygote. About 25% of all fertilized egg cells are aborted spontaneously within the first few weeks, usually without the mother even knowing she was pregnant. Of the zygotes that survived the first 3 to 4 weeks after fertilization, a further approximately 30% are aborted spontaneously.
After jumping into the basics of prenatal development, I want to turn our focus into a deep dive of genes, DNA, and chromosomes.
Now let's focus on chromosomes, genes, and DNA. A new life begins as a zygote containing 46 chromosomes. A chromosome is found inside the nucleus. This is the center of every cell in the body. Each person contains 23 pairs of chromosomes from the mother and from the father. So every cell in the body has 23 chromosomes. Now genes. Each chromosome is made up of many genes. Carry all human hereditary characteristics. Humans have about 20,000 to 50,000 of genes. Scientists do not yet fully understand how genes work or interact with each other. DNA. So this is a deoxyribonucleic acid. It is the chemical substance of heredity, the material that stores genetic information. DNA carries the recipe or the blueprint for how cells should function. It looks like a double spiral twisted letter called a double helix. About 0.1% of DNA is responsible for the physical and psychological differences between people. DNA strands are all small, that about 5 million could fit through an eye of a needle. The next part is dominance and recessivity. How traits are determined. Each pair of chromosomes carries 2 pairs of genes that are responsible in each specific characteristic. One gene comes from the mother and the other gene comes from the father. When 2 genes are identical, the child will show the same characteristics. So for example, if both parents pass down the gene of brown eyes, the child will come out with brown eyes. When genes are different, so sometimes you receive different genes in which one eye is brown and the other eye is blue, for example. This shows genetic dominance and recessivity. Dominant genes. A dominant gene is the one that shows its effect even if the other gene is different. So a gene that determines your physical trait, this is called the phenotype. And a gene that determines your genetical trait, this is called a genotype. Recessive genes. A recessive gene is the one that doesn't show its effectiveness. Its characteristics stay hidden in a person's genotype, genetic makeup, but it can still be passed down for future generations.
Now to further elaborate on this, I'll be taking over. Most characteristics are determined through the effect of or interaction between many genes. This is called polygenic inheritance or multifactorial inheritance. Characteristics such as temperament and intelligence are known as polygenic characteristics. To further elaborate on genotype and phenotype, well, first I have to look at the difference between the two. The genetic information or makeup of an individual found in the individual cell. Phenotype is the observable physical traits based on the expression of the genotype. G is genetic and pH is physical. Unfortunately, during the prenatal stage, there is such a thing called congenital disorder. First, we must understand the difference between inherited disorders and conception genetic disorder. Inherited disorder, that is disorders that are inherited from one or both of the parents. Hyperactivity disorder and major depression tend to run in families and are believed to have a strong inherited basis. Conception genetic disorder, that is the result of an error that happens during the conception process. For example, Down syndrome. Any external factor or process that has a negative effect on the normal development of an unborn child. These are known as prenatal environmental influences or teratogens. Teratogens are agents that can induce or increase the incidence of birth defects in a developing child. Time of exposure, genetic vulnerability, and the degree or intensity of the factor that plays a role are all considered prenatal environmental influence. More damage can be done at certain phases of development, and more damage can be done to certain genes, and more damage can be done when more damage is done. The point is to stay away from any teratogen when you are pregnant, because not everybody knows that they had a teratogen interaction. The damage is not always evident at birth, and each teratogen has a different effect on the unborn child. The damage is selective. Examples of prenatal environmental factors that affect the development of a child negatively are age, nutrition, infectious diseases, medications and drugs, and even the emotional state of the mother. Now, I truly do hope that you learned something from the prenatal development. And as always, goodbye. And a special thanks to everybody who made this video possible. We couldn't have done it without you. Love ya.